and welcome to Spotlight on Antibodies, a podcast from BioIntron where we bring you the latest antibody industry trends. This month in January, we'll do a deep dive into last year's novel antibody drugs, G-protein coupled receptors, and fusion proteins. In 2025, the antibody drug field had a particularly strong year. Worldwide, 21 novel monoclonal antibody therapies were approved for the first time. Compared with earlier years, 2025 reflects a clear expansion in antibody modality and therapeutic focus. Alongside conventional monoclonal antibodies, we saw approvals for bispecifics, antibody drug conjugates, long-acting formulations, and Fc-engineered molecules. These therapies span oncology, autoimmune disease, rare disorders, and infectious disease prevention, showing how broadly antibody technologies are now being applied. One of the most notable scientific trends this year is a shift in target selection. Rather than focusing primarily on tumor-associated antigens, many newly approved antibodies act on immune regulation pathways. More than a third of approvals target soluble immune mediators, including cytokines, complement components, and antibody receptors. Several antibodies approved in 2025 target inflammatory cytokines involved in chronic immune disease. These include therapies directed against IL-5 for asthma, IL-beta for gout, and IL-23 or IL-12 for psoriasis. Another example is sibeprenlimab, which targets APRIL and was approved for IgA nephropathy, an area with historically limited treatment options. Beyond cytokines, antibodies acting on the complement and coagulation systems also featured prominently, with approvals for conditions such as transplant-associated thrombotic microangiopathy and hereditary angioedema. We also saw progress in antibodies that regulate immunoglobulin biology, including FcRn-blocking antibodies, designed to reduce pathogenic IgG levels in autoimmune disease. While these mechanisms are biologically complex, most of the approved drugs still use standard IgG1 or IgG4 formats. In some cases, Fc domains are engineered to adjust immune activity or extend half-life, but the underlying antibody platforms remain well established and clinically validated. Another important shift in 2025 is the therapeutic area distribution. Oncology no longer dominated new monoclonal antibody approvals, as only 1/3 of the approved drugs were for cancer, including immune checkpoint inhibitors, bispecific antibodies, and several ADCs targeting proteins such as HER2, EGFR, and c-Met. In contrast, the majority of approvals actually addressed autoimmune and inflammatory diseases, along with several antibodies developed for infectious disease prophylaxis. This reflects the growing role of monoclonal antibodies in precise and durably modulating immune function, particularly in chronic conditions. The geographic distribution of approvals is also interesting. More than half of the novel mAbs approved in 2025 received their first approval in China, with fewer first-in-world approvals occurring in the United States and Europe. Notably, China's approvals included complex modalities such as ADCs, cytokine blockers, and immune checkpoint inhibitors, highlighting both strong local innovation and a rapidly maturing regulatory environment. Finally, several approvals this year highlight progress in long-acting and preventative monoclonal antibodies. Through Fc engineering and structural optimization, some antibodies now support dosing intervals of 6 months or longer. These long-acting therapies are being used not only for chronic inflammatory disease, but also for prevention of infections such as COVID-19, RSV, and tetanus. Overall, the monoclonal antibodies approved in 2025 reflect a field that is expanding beyond oncology, embracing immune modulation, advancing long-acting design strategies, and becoming increasingly global where innovation takes place. After looking at how antibody approvals expanded in 2025, it's worth zooming in on one area that's drawing growing attention: G-protein coupled receptors, or GPCRs. GPCRs make up the largest family of druggable membrane proteins in the human genome. There are more than 800 GPCRs, and they regulate critical physiological processes ranging from immune signaling and metabolism to cardiovascular and neurological function. Small molecule drugs targeting GPCRs account for roughly 1/3 of all FDA-approved medicines. Yet despite this long history, only 3 GPCR targeting antibodies have been approved to date. That gap has made GPCRs one of the most challenging and potentially most rewarding frontiers for antibody drug discovery. Historically, GPCRs have been difficult antibody targets for several reasons. Many receptors are expressed at very low levels on the cell surface. Their extracellular domains are often small, and the receptors themselves are highly dynamic, adopting multiple conformations depending on signaling state. In addition, many GPCRs have unknown ligands or poorly understood signaling pathways, further complicating drug discovery efforts. However, recent advances are beginning to change this landscape. Improvements in membrane mimetic expression systems, functional screening technologies, structural biology, and artificial intelligence are collectively overcoming barriers. One important shift has been toward function-driven discovery. Traditional antibody screening approaches prioritize binding affinity, but for GPCRs, binding alone is often not enough. To identify antibodies that actually modulate receptor signaling, researchers are increasingly using functional screens designed to detect agonists, antagonists, and allosteric modulators. For example, Abalone Bio has developed a platform capable of functionally screening hundreds of millions of antibody variants. In collaboration with Pfizer, the group is working on activity modulating antibodies against an undisclosed GPCR target, using AI-trained models built on large-scale functional datasets rather than relying solely on structural information. At the same time, AI-driven antibody design is playing an expanding role. A collaboration between Nxera Pharma and Antiverse is focusing on designing de novo antibodies against GPCR targets using generative artificial intelligence. In this approach, machine learning models generate epitope-specific antibody libraries that are pre-tuned to distinct receptor conformations, enabling more targeted and efficient identification of functional antibodies. Structural biology has also advanced rapidly. Techniques such as cryo-electron microscopy and X-ray-free electron laser crystallography have revealed previously inaccessible GPCR epitopes, including conformational binding pockets and ligand access channels. These insights support structure-guided design of antibodies that act as orthosteric or allosteric modulators, and even enable engineering of biased agonists that selectively activate specific signaling pathways. AI-assisted protein modeling is now further accelerating this process by predicting GPCR antibody interactions and optimizing antibody conformations for dynamic or partially buried epitopes. These tools are especially valuable for so-called orphan GPCRs, where antibodies can function not only as therapeutics, but also as probes to help define receptor biology. Beyond conventional monoclonal antibodies, new modalities are expanding what GPCR targeting can look like. These include peptide antibody fusions, such as GLP-1 Fc constructs that enhance half-life and tissue localization, as well as bispecific antibodies that enable dual targeting in oncology. GPCRs are also being explored in antibody-drug conjugates and CAR T-cell therapies, particularly for receptors such as CXCR4 and GPRC5D. Together, these approaches move GPCR drug development beyond simple agonism or antagonism, opening possibilities for receptor internalization, cell-specific cytotoxicity, and pathway-selective signaling. With only 3 GPCR targeting antibodies proved so far, the integration of functional screening, structural biology, membrane engineering, and AI-driven design has the potential to unlock dozens of new therapeutic targets. To close out this episode, we'll turn to another area in antibody research, fusion proteins. Fusion proteins are engineered molecules that combine functional domains from two or more proteins into a single polypeptide chain. In antibody drug development, this often means pairing an immunoglobulin framework with additional components, such as cytokines, enzymes, or synthetic peptides, allowing researchers to create molecules with entirely new biological properties. Antibody fusion proteins offer several pharmacological advantages. By incorporating an Fc domain, these molecules benefit from FcRn-mediated recycling, which extends serum half-life. At the same time, fusion formats can improve molecular stability, enhance targeting precision, and enable controlled delivery of potent payloads. As a result, they are being explored across oncology, autoimmune diseases, infectious diseases, and neurodegeneration. The field has evolved significantly from early Fc fusion drugs, such as etanercept, which helped establish fusion proteins as a viable therapeutic class. More recent efforts focuses on rational molecular design, aiming to overcome challenges including cytokine pleiotropy, systemic toxicity, and short in vivo persistence. Two major trends now define the landscape. The first is modular immunocytokine design. Antibody cytokine fusion proteins are increasingly engineered to bias receptor signaling, restrict activity to target tissues, and extend half-life through intramolecular configuration. The second trend is payload diversification. Beyond cytokines, fusion partners now include amyloid reactive peptides, enzymes, toxins, and immune checkpoint modulators, significantly broadening the therapeutic scope of fusion formats. Fusion proteins are also emerging as tools in antibody discovery itself. A recent study described a fusion-based immunogen strategy designed to address one of the field's long-standing challenges, generating antibodies against unstable or transient protein complexes. By genetically fusing the immune regulatory proteins BTLA and HVEM into a single construct, researchers were able to stabilize the complex and successfully generate monoclonal antibodies capable of detecting both individual proteins and their complex form in live cells. This approach offers new opportunities for targeting conformational epitopes implicated in diseases such as lupus and lymphoma. Another promising application is amyloid diseases, where fusion proteins may enable broader therapeutic coverage. An antibody fusion protein named AT02 combines a humanized IgG1 with a pan-amyloid reactive peptide. This design allows it to bind multiple amyloid subtypes, as well as associated heparan sulfate glycans, acting as an opsonin to enhance phagocytosis and complement activation. Preclinical studies demonstrated strong in vivo binding to amyloid deposits, supporting its ongoing evaluation in clinical trials for systemic amyloidosis. By avoiding the need for subtype-specific antibodies, this strategy could simplify development across heterogeneous amyloid disorders. In oncology, fusion proteins are being engineered to deliver multiple cytokines simultaneously to enhance immune synergy with the tumor microenvironments. Recent work on trifunctional antibody cytokine fusion constructs showed that cytokine positioning within the molecule has a major impact on activity. Constructs combining IL-15 with IL-21, particularly those using scFv-based antibody domains, significantly enhanced CD8 T-cell cytotoxicity and interferon gamma production compared with single cytokine formats. These findings highlight how spatial organization within fusion proteins can determine therapeutic potency while limiting off-target effects. Overall, fusion proteins are redefining what antibody-based therapeutics can achieve. Advances in structural design, target biology, and payload integration are converging to create a diverse pipeline of multifunctional agents. As understanding of immune regulation and disease mechanism deepens, fusion proteins are expected to play an increasingly central role in precision medicine, enabling more targeted, durable, and biologically sophisticated therapies across a wide range of indications. And that's all for this month's Antibody Industry Trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.